All right. So welcome to the fifth lecture of physical biology module, and let's start by having a quick look at what we did in the last lecture. So in the last lecture, we were talking about diffusion, and one of the things that we did in the last lecture was that we derived the diffusion equation by combining the Fick's law and continuity equation. So diffusion equation is an equation which describes the process of diffusion. It connects the rate of change of concentration with time to rate of change of concentration with space. Okay, and what we saw that diffusion equation was although very simple, it had similarity to many other known equations like Schrodinger equation, heat equation, wave equation, but it is one of the hardest equation to solve, and therefore we also did not solve the diffusion equation in the last lecture. What we did, we looked at the solution of the diffusion equation in a very special case, which was free diffusion from a point source, which means something was very much concentrated at one place and then it was released at t equal to 0. What we found that if we have something like that, then the concentration profile spreads like a Gaussian function with time. And one of the important thing that we saw in the last lecture, that when something is moving ballistically from one point to the another point with a certain velocity v, constant velocity v, then in that case the displacement x is proportional to t. However, if something is going from one place to the other place by the process of back and forth movement like diffusion, in that case the displacement x does not grow as t, but it grows as t to the power half. Okay, and a consequence of that is that if we compare the displacement of a particle which is undergoing ballistic motion versus a displacement of a particle which is undergoing a diffusive motion, then over longer time scales the distance traveled by a particle which is undergoing diffusive motion is will be much more smaller, and that's why we say that diffusion is a very slow process. And what we found in the last lecture, that diffusion is such a slow process that it will take about 30 millennia, 30,000 years to go an organelle from my head to toe. Okay, so we cannot definitely survive using the mechanism of diffusion, and that's why our cells use motor proteins such as kinesin and dynein, which move along microtubules, and unconventional myosin motors, which move along actin filaments to transport things from point A to point B, utilizing energy which is obtained from the hydrolysis of ATP. Okay. After that, we looked at the contribution of Einstein to the field of Brownian motion, and we derived the Einstein relation. So Einstein relation is given as gamma d is equal to kVt, and tells you that in thermal equilibrium, the product of diffusion constant and drag coefficient is a constant quantity, or in other words, they try to balance one another. So if one of them decreases, the other one has to increase. And the significance of this Einstein relation was that one could use the Einstein relation in year 1905, and one could calculate the Avogadro's number using a simple microscope and a stopwatch. Okay, and his derivation of Einstein relation and his theory of Brownian motion gave a very strong support to molecular kinetic of theory of heat, which was based on the assumptions that a matter consists of particles, which were later called as atoms and molecules. After that, we discussed the reaction diffusion system. So diffusion in itself is very interesting, but when it gets combined with reaction, it becomes even a much more richer system. And I told you that Alan Turing, whom we know as a very good computer scientist, a very good mathematician, he was also a pioneer in the field of reaction diffusion system. And this was something which was used by the scientists in past to understand the pattern formation in nature. All right. So what we are going to do in this lecture, we are going to talk about protein and their structures. So in the last lecture, I talked about microtubules and actin filaments, right? So I told you that microtubules are like long range highways which go from cell center to cell periphery. Similarly, actin filaments are like short filaments, like local roads, which are mostly near the cell surface. Okay. So if you look at these microtubules, these are filamentous proteins. But these proteins, which have size of few microns, okay, these filamentous proteins are polymers of protein monomers. So for example, microtubule is a polymer of alpha and beta tubulin. So alpha and beta tubulin make a dimer which is called alpha beta tubulin heterodimer, and that constitutes the microtubule. Similarly, the filamentous actin filament is a polymer of G-actin or globular actin protein. Okay. So important thing is that most of the cytoskeletal filament are polymers. So although the length of the microtubules is few microns, the size of these alpha and beta tubules is of the order of nanometers. For example, about let's say 4 nanometers. Okay. And if you look at each of these proteins, so let's say alpha protein and alpha monomer and beta monomer. So this alpha beta heterodimer, if you look at this, it consists of a large number of amino acids. So the total number of amino acids that are there in a tubulin heterodimer, there are 863 amino acids, and alpha tubulin itself has 436 amino acids and beta tubulin has about 427 amino acids. And if you look at the sequence, this is how you'll find the sequence of amino acids. Okay, so each of the letters corresponds to one amino acid. Okay, so there are total 436 in alpha and 427 in beta. And normally people try to show the protein structures using these kind of cartoons. Okay, where helices are shown as squares, then there are these flat structures which are beta sheet, and anything which is shown with the loops is unstructured. Okay, now this is how this cartoon representation of the protein looks like, but actually if you look at the protein structure, it consists of thousand of atoms. Okay, for example, this alpha beta heterodimer consists of about 8,000 atoms. So even one heterodimer can have 8,000 different atoms. Okay, so this is the complexity of the protein molecule that we have. However, in the simplest view, protein is consisting of amino acids, and all of these amino acids are made from atoms, right? And even a small protein can have thousands of atoms. Okay, so this is what I was telling you, that if you look at the protein structure, there is a primary sequence which is just the sequence of amino acids. Then they undergo local folding, which makes alpha helix and beta sheets, and they finally make tertiary structure of the protein. If you look at the functional structure of the protein, that is often quaternary and consists of many domain like structures. And all of these proteins which are there inside our body, thousands of these different types of proteins, all of these proteins are made from only 20 different types of amino acids. Okay, so if you go back to the previous slide, this entire protein is made from only 20 different types of amino acids. Similarly, this monomer is also made from only 20 different types of amino acids. So you find only not more than 20 different type of letters. Okay, now these are the proteins which carry out all kinds of functions in our body. For example, they do signaling, they do the transport of ions across the cell membrane, which is responsible for my heartbeat, which is responsible for nerve conduction. Okay, they catalyze a lot of reactions. They power the movement of the muscles inside the body. For example, when I'm talking to you, moving my head, moving my limbs. In all of those processes, they are the myosin motors which are sliding the actin filaments. Similarly, they provide the structural support like microtubule and actin filaments. They can regulate the things. Important thing is that all the functions inside the body are carried out by the proteins, and these proteins, these 1,000 different types of proteins which works inside our body, are made from only 20 different types of amino acids. Okay, and these 20 different types of amino acids get arranged into different sequences that give rise to different shapes to this protein, and protein perform their function by folding into different shapes. So proteins are sequence of amino acids, and there are not more than 20 amino acids which make all kinds of natural proteins. Okay, we have 26 alphabets in English. If I give you a sequence of 26 alphabets on a page, okay, by looking at the sequence of the letters, you can tell me what I have written on the page. Okay, what is the structure which is there, whether it's a song, poetry, speech, or something else. But if I give you a sequence of amino acids like this, by looking at sequence of, let's say, even 20 amino acids, you cannot tell me what will be the three-dimensional structure of the protein. So given a sequence of amino acids, can we predict the structure of the proteins with 100% accuracy? Is still an unsolved problem, because we don't know the exact rules according to which protein fold, and people try to do computer simulation to study, to basically predict the structure of the protein. So there are ways of doing it, but we still don't know the exact rule according to which protein fold into three-dimensional shapes. So what we are going to do in this lecture, we are going to understand the problem of protein folding by developing some toy models. And in this particular lecture, I will use a toy model which is called HP model of protein folding. Okay. So this is just a revision of what I told you, that there is a primary sequence of when the protein is being made, that there is a primary sequence which is just a sequence of amino acids that folds into secondary structures like alpha helix and beta sheet, that gives rise to tertiary structure which consists of alpha helix, beta sheets and loop like regions, and quaternary structure can consist of many such domains. Okay. Now if you look at the proteins are polypeptides, because all of these amino acids are connected together by peptide bonds. So if you look at the structure of the amino acids, most of the all the amino acids have a common structure. What is different between different amino acids are the side chains, and these unique side chains give the unique properties to each of the amino acids. So if you have to look at the simple picture of protein, you can think of that there is a common backbone to which these different types of side chains are attached. Okay. So what we'll do here, as I said, that all different 1,000 types of proteins are made from 20 different amino acids. And here we'll try to understand the problem of protein folding by having a very simple model of protein. So I will say that, okay, there are not 20 different types of amino acids. Let's assume that all known amino acids can be broadly classified into two classes, hydrophobic and polar. So hydrophobic are the ones which would not like to be in contact with water, and polar ones are the ones which would like to be in touch with water. So for me, there are not 20 different types of amino acids. There are only, let's say, two different types of amino acids, hydrophobic and polar. Polar ones would like to be in contact with water, and hydrophobic ones would like to be away from the water. Now suppose I have a polypeptide chain, okay, which is consisting of some hydrophobic and some polar amino acids, and let's say I put this chain into water. Okay. Now based on this information, what can you say about the presence of polar amino acids and hydrophobic amino acids when I throw this polypeptide chain into water? In what kind of shape it would fold? Let's say if this protein starts to fold when I put it into water, what kind of shape it would acquire? Anyone? Anyone? Please raise your hand. So let's say I throw this polypeptide, which is consisting of hydrophobic and polar amino acids, into water, and it starts to fold. What kind of inference you can make about the presence of polar and hydrophobic amino acids when this polypeptide is folding into water? Where you will find the polar amino acids and where you will find the hydrophobic amino acids?
Sir, they will try to fold like that. Hydrophobic part will be inwards and hydrophilic part will be outwards.
Okay, very good. So the polar ones would try to be on the surface, and hydrophobic ones will try to be inside the core region. That means inside region, and that is the only way they can minimize the energy, right? Okay, very nice. All right, so this is what I have told you so far, that proteins are sequence of amino acids, and what we are trying to understand, if we are given a sequence of amino acids, can we predict the structure of the protein? Now the problem is that if you apply the classical mechanics to this problem, then classical mechanics will tell you that protein would prefer a structure or conformation which minimizes energy, right? So generally we say that any system tries to go to its minimum energy state, right? However, this is not true when thermal fluctuations are present. And we know that when we are in the microscopic world, we cannot ignore the thermal fluctuation. In the macroscopic world, we can ignore the thermal fluctuation, but when we are in the microscopic world, this thermal energy starts to compete with the other forms of deterministic energy, and therefore we cannot ignore the thermal energy, and we have to take into account the thermal energy or thermal fluctuations. So when the thermal fluctuations are present, then in that case system does not tries to minimize its energy. What system tries to minimize is something called free energy. Okay, so in presence of thermal fluctuation, a system tries to minimize its free energy. Now my question for all of you is that how many different types of free energies you know about? What are the different types of free energies you know about? Anyone? So what I'm telling you that in presence of thermal energy, system tries to mini. क्या बहुत काम आने वाला है. So what are the different types of free energy you know about? Anyone? What are the different types of free energy you have heard of? Please raise your hand. What are the different types of free energy you know about? Have you heard of free energy before or not? Yes. Athar?
Gives free energy.
Okay, gives free energy we know, and that is given as G is equal to H minus Ts. Any other kind of free energy you have heard about? So there is another kind of free energy which is called Helmholtz free energy, which is given as A is equal to U minus Ts. Okay. Now, if you look at this gives free energy, H is nothing but it is U plus PV minus Ts, right? H enthalpy is U plus PV. So whenever we are talking about the protein folding, there is zero change in the pressure of the protein. Okay, and especially this term is almost zero, and therefore in the case of protein folding, whenever I'm talking about the free energy, I'm actually talking about the Helmholtz free energy. So for me, the expression for the G will be U minus Ts. Okay, so in presence of thermal fluctuations or thermal energy, a system tries to minimize the free energy, and that is the case in the microscopic world. Okay, so a system tries to minimize the free energy, that is G has to go down. And in how many ways we can reduce the G? Either I decrease the average energy of the system, or the other thing I can do, I increase the entropy of the system. If I increase either the entropy of the system or decrease the average energy of the system, in both of these cases, the free energy will go down, and we know that any system tries to minimize the free energy, right? So, question is, what is this entropy? Now, all of you have read about entropy before, maybe at the school level. So, I would like to know from you what is entropy. Anyone? What is entropy? Everyone has probably studied the entropy. Please answer. We have about 130 students online right now. What is entropy? Please raise your hand and TS will unmute. What is entropy? Yes. Please unmute the students who have raised the hand. is?
It's the measure of degree of randomness.
Okay, it's the measure of degree of randomness present in the system. Any other definition? Second student who has raised the hand.
It was the same.
Sorry.
I was going to say the same.
Okay, so whenever I ask the question, what is entropy, I get the answer. There is one more hand. Okay, so I get the answer that entropy is a measure of disorder present in the system. Entropy is a measure of randomness present in the system. Okay, so these are the vague definitions of entropy. These are not the exact definitions of the entropy. If you tell me that it is the measure of randomness present in the system, disorder present in the system, my next question will be, how do you compute it? How will you measure the randomness or disorder present in the system, right? So next question is how to calculate entropy. So any other definition of entropy, All right. So whenever we ask about the entropy, I mean, we hear these vague definitions. Yes, somebody raised the hand. Please go ahead.
Entropy can also be interpreted as energy which cannot be used to perform useful work.
No, probably not a correct one. Okay. Okay. So whenever we ask the question that what is entropy, we get the answer that it's a measure of disorder present in the system, it's a measure of randomness present in the system, but those are the vague definitions of the entropy. Okay, so what I'm going to tell you is the exact definition of the entropy. Okay, so let's look at that. Okay. So what is written here, that entropy is a measure of microscopic degeneracy of a macroscopic state or macro state. In other words, entropy can be computed by counting the number of possible microscopic arrangements or states, which are also called microstates, for a given macroscopic state or macro state. Okay, so what is being told to you is that entropy is a measure of microscopic degeneracy of macroscopic state, and you can calculate the entropy of a macroscopic state by counting how many microscopic states are there. So you can see that on this slide, I'm using two words. One of them is microscopic state or micro state. The other one is a macroscopic state or macro state. And it tells you that if you can count the number of micro states, then you can calculate the entropy of the system in that particular macro state. Okay, so let's try to understand the meaning of macro state and micro state. Okay, so let me go back to the next slide. Okay, so here let's take the example. I'm also turning on my video. So here what you see, that I have an example of protein which can exist into two states. One of them is a open state, the other one is a closed state, right? Now this is a protein which is in the open state. Okay, now even in this open state, although the overall state is an open one, this protein is consisting of a large number of amino acids. Okay, in turn this protein is also consisting of a large number of atoms, and if I have this protein in open state at a finite temperature, all of these atoms, because of the thermal energy, will keep moving all the time. So even if the protein remains in the open state, each of the atoms of the protein can be in different places at different times. Okay, so overall the state looks to me like open state, but each of these state will consist of many microscopic arrangement of atoms. Okay, so in this case the open state will be called the macro state, and all the different atomic arrangements that correspond to this particular macroscopic state will be called the microscopic states corresponding to the open macroscopic state. Similarly, when the protein is in the closed state, no ion can pass. So although overall the state looks to me like it's a closed state, but if you look at this closely, even in the closed state, all the atoms will keep changing their position because of thermal fluctuation. So even in this closed state, there can be different possible arrangement of the atoms, and each of these different possible arrangement of atoms in the closed state will give rise to different microscopic state that will correspond to a given macroscopic state. Okay, let me go to the iPad here just for a second. Okay. So what we are trying to understand is the meaning of macroscopic state. So we have a macroscopic state. This is also called in short macro state. Okay, so let's say there are two states, open and closed. Okay, but each of these open and closed macroscopic state can consist of many possible microscopic states or micro state. Okay, so each of the open state can consist of many possible atomic arrangements, and therefore each of these states can consist of a large number of possible configurations which will differ in energy from one another in a slightly we will have slightly different energies. Okay, let's look at another example. Okay, so again I'm turning on my video, please look at. So right now my hands are in the straight state, right? My hands are straight. So this is one state, straight state. This state will be called the straight macroscopic state. But even if I'm in the straight state, I can move my fingers in different ways. So although the entire arrangement looks to you like a straight one, but each of even this global macroscopic state, each of my finger can take different possible orientations. Okay, and therefore, and you know that the energies of different configurations will be different here, right? So the same thing also applies to the this channel protein example. So each of the atomic arrangements will correspond to a different microscopic state. For example, I can create a large number of microscopic states here. Okay, now this is the folded state of my hand, and even in the folded state, my fingers can take different possible configurations. So if you look at this globally, it is a bent state, but even this bent state can consist of many different possible arrangement of the fingers. Okay, so this state or this state are like macroscopic states. Okay, straight and bent, but each of these states can consist of different possible finger positions. So same thing applies to the protein. Now you think about this protein. Even if the protein is in the open state, it consists of many possible arrangement of atoms, and each of these correspond to different arrangement of atoms, and therefore the energies of the different configurations will be slightly different. So all of these will be called the microscopic states corresponding to the open macroscopic state. Similarly, if you look at the closed one, again it can consist of many possible atomic configurations, and energy of the protein in different atomic configuration, different configurations of for different configurations of atoms will be slightly different, and these ones will be again called the microscopic states corresponding to this one, closed one. Okay, so this is the meaning of the macroscopic state and microscopic state. So one given macroscopic state can have many possible microscopic states, okay, which can differ in energy from one another. All right, is this clear? Any questions on macroscopic state and macroscopic state? I hope this is clear to everyone. Is the meaning of macroscopic state and microscopic state is clear? What we mean by macroscopic state and microscopic state. All right. So if this is clear, then what is being told to me about the entropy? That I can calculate the entropy of a system by calculating how many microscopic states are there. So for example, if I ask you, let's say this is the open microscopic state and it has total 15 possible microscopic states, and the other one, closed one, has only 10 different possible microscopic states. Which one of these two will have higher entropy? Anyone? The open one will have a higher entropy, or the closed one, closed macroscopic state, will have a higher entropy? Please raise your hand. Yes, go ahead.
The open one.
Sorry?
The open one will have higher entropy.
Open one will have higher entropy. That is a correct answer. But why? What is the reason for that, Nayantara?
Because there are more possible microstates.
There are more number of microscopic states, and so here what is happening, that here if you look at this system, open state, here system can jump into 15 possible different states. On the other hand, if you look at the second system can jump into only 10 different possible states. So in that sense, the randomness which can be created in the first one is much larger than the randomness that can be created in the second one, right? And this is why the first one will have a higher entropy. All right. So by counting the number of microscopic states corresponding to a given macroscopic state, we can calculate the entropy. And entropy is given by the formula S is equal to Kb ln W, where W is the number of microstates. Okay. And this entropy is entropy of the, this is the entropy of a macroscopic state. So if someone is asking me a question, what is the entropy of open macroscopic state is, I will count how many different possible microscopic arrangements are there or microstates are there, and then I will apply this formula to calculate the entropy. Okay. Now this is something which is called thermodynamic entropy, and this was given by famous physicist Boltzmann. Okay. Now let's go back to slides. So if you ever get a chance to visit Vienna, this is the gravestone of Boltzmann, and see what is written here, S is equal to K log. So by counting the number of microscopic arrangements or microstates, we can calculate the entropy, and this formula for entropy is called Boltzmann entropy because it was given by Boltzmann. Okay. Now let's again go back to the iPad. All right. So now suppose I have a system. So now we understand what is the meaning of a macro state is. Okay. So it can be an open state and a closed state, or it can be, let's say, a straight state or a bent state. And we know that each of them can consist of many possible microstates. Okay. For example, open one can consist of a large number of possible microstates. Similarly, closed one or bent one can also consist of many possible microstates. Okay. So these are called microstates. Okay. Now let's say this is a state 1 with energy E1. This is state 2 with energy E2. This is state I with energy EI. This is state N with energy En. Similarly, let's say this is state 1 dash with energy E1 dash, state 2 dash with energy E2 dash. Let's say this is state J with energy EJ, and this is, let's say, state M with energy EM. Okay. Now suppose you are given a system. There are two possible macroscopic state, open and closed, and each object can consist of a large number of possible microstates. Let's say open consists of total n number of microscopic states, and the closed one consists of n number of possible microscopic states. And now suppose somebody is asking a question, what is the probability that you find the system in this particular state? What will be your answer? What will be the probability of finding the system in state I? How will I get it? Any idea? Which law I will apply? Anyone? Please raise your hand. How do I get the probability of finding the system in a particular state I? Recall lecture 3. Yes, Athar.
We divide the energy of that particular state by the summation of all energies.
No. Any other answer? Which law we have to apply? Yes.
Boltzmann law.
Huh?
Boltzmann law.
We can apply the Boltzmann law. So if I have to find out what is the probability of finding the system in a particular state I is, Boltzmann law tells me that Pi will be proportional to, or Pi can be written as e to the power minus Ei by kBt divided by Z. Okay. And if you are writing the Z, if the overall description of the system is given, okay, all the states are described clearly, then this Z will be sum of has to be a sum over all of these energy states. All right. Similarly, if somebody is asking you a question, what is the probability of finding the system in this state J is, again you can apply the Boltzmann law. You can say that Ej is equal to e to the power minus Ej upon kBt divided by Z. All right. Now suppose instead of asking you probability of finding the system in a particular microscopic state, somebody is asking you a question, what is the probability that the system will be found in open microscopic state? What will be your answer? Anyone? So if let's say I'm not interested in finding the probability of a system in a particular microscopic state, I am interested to find out what is the probability that the system will be found in open macroscopic state or it will be found in closed macroscopic state. How do I find it? Anyone? Any idea? Please raise the hand quickly so that we can move ahead quickly. How do I do this? Any volunteer? Please give me an answer even if it is wrong.
Take the sum of probabilities from P1 to P.
Very nice. So, if somebody is asking me a question, what is the probability of finding the system in the open microscopic state is, I can take all the sum over Pi's, right? Similarly, if I take the sum over all the Pj's, I get the probability of finding the system in a closed macroscopic state, okay? So, if somebody is asking you a question about a microscopic state, you can apply the Boltzmann law. And you use the energy of particular microscopic state to find out the probability for that particular microscopic state. However, if somebody is asking you that what is the probability that system will be found in a macroscopic state X, then again you can use the Boltzmann law. However, the difference is that here instead of having the energy of the state, what you will have here you will have the free energy of the macroscopic state divided by. Okay, so for example, if I have to find out what is the probability of finding the system in open macroscopic state is, I can find it out by calculating what is the what is the free energy of the open macroscopic state is. Okay, so important thing for you to remember is that if the question is about a microscopic state, you can use the Boltzmann law. You have to use the energy of that particular microscopic state. However, if the question is being asked about the probability of a macroscopic state, then in that case, you have to replace this energy by the free energy of that particular macroscopic state. Okay, you can still use the Boltzmann law, but the energy has to be replaced by the free energy. And here you can see that any macroscopic state which will have a lower free energy, it will have a higher probability, right? Okay, so I hope all of this is clear to you. Let's go back to the slides. Okay, so this is what is written here, that if the question is being asked about the probability of finding a macroscopic state X, then it is given by summation over all the PIs which correspond to the macroscopic state X, and it can be shown that this will be equal to 1 upon Z e to the power minus Gx by kVt. Okay, so this is the similar to the formula for the microstate, but here the energy is being replaced by the free energy of the macroscopic state. All right, now how do we get this relation G is equal to minus kVt ln Z? That derivation is given here on this page. You can look at this derivation, and here we are using another alternative formula for the entropy. This formula for the entropy is called Shannon's entropy. The formula which is S equal to kV ln W is called the Boltzmann entropy, and this formula is called the formula for the Shannon's entropy. This is something which is used very heavily in electronics and communication engineering. One can actually show that this formula of Shannon's entropy is actually equivalent to the formula for the Boltzmann entropy if you replace the K by kV, and this is one of your tutorial problems, so please look at this. So this is the summary of what I have told you so far, that if we have a protein, then it can exist in many structural states which are like macrostate, open, closed, straight, bent, and each macroscopic state or macro state consists of many possible microscopic arrangements which are also called microstates. Okay, and what I have told you that the macro state which has minimum free energy will be preferred because of this dependence of Px on G. Okay, so we have looked at the formula for the free energy. Okay, gives free energy. We have looked at the relationship between G and the partition function, and we have also looked at the formula for how to calculate the entropy. And what we have finally found, that probability of finding a macroscopic state X is given by Px is equal to 1 upon Z e to the power minus Gx upon kVt, where Gx is the free energy of the macroscopic state is. Okay, so now with all of this background, let's try to look at one simple model of protein folding, which is the HP model of protein folding. Okay, so as I said in the beginning, that for me, I will consider that all known proteins can be broadly classified into two classes. Okay, hydrophobic and polar, and what we are going to discuss is a simple model of protein folding, which is called HP model of protein folding, because here we are considering that a protein is consisting of only two different types of amino acids, hydrophobic and polar. Hydrophobic ones would like to be away from water, and polar ones would like to be in touch with water. So what is given to you in the problem, that consider a protein consisting of 8 amino acids H and P. The sequence of amino acids is H, P, H, 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 P. Further, it is given that suppose this protein can fold into 5 possible structures such that energy increases by epsilon for every contact of H with either P or solvent molecule. Okay, so what is given to you in this problem, let's say that this protein can fold only in 2D, and these are the 5 different possible configurations in which it can fold. What is also given to you, that energy will increase when H is right next to a P. So if H makes a contact with a polar amino acid, that means if H comes within the bonding distance of a polar amino acid, then there is a increase in energy, or H comes into the contact with water or solvent molecule, then there is a increase in energy. Okay. And what is given that suppose these structures can be broadly classified into two macroscopic states, open and packed. So I said that these are the 5 possible structures. Since these ones look much more open, I am saying that this macroscopic state is being called as open. Okay, because protein looks like it's much more open, and last two are being classified as packed because everything is squeezed into 2 layers. Okay, now question which is being asked, that what will be the structural state of the protein at temperature T, whether it will be found in open state or packed state, or which state will be preferred, whether it will be open state or it will be the packed state which will be preferred. Okay, so we have to find it out. Now how do we proceed with this problem? So we have to find out in which macroscopic state I will find this protein. How do I do that? Please turn off your video. Om Gadade.
So my video is off.
Yeah. Okay, so how do I proceed with this problem? Any suggestion on how should we proceed with this? We have to find out in which state the protein will be found. Okay, or let's say which macroscopic state has higher probability, because if it has a higher probability, then it will be found in that state, right? So how do we proceed with this problem? What do we need to calculate? Any thoughts? Please raise your hand. This has to be an interactive lecture. Please give me answers. Even if it's wrong, it's fine. Anyone? What do we have to find out to say whether it will be found in packed state or open state? What should we calculate?
The probability of each.
Probability of each of the states, right? And in order to calculate the probability of each of the macroscopic state, what do I need to calculate? Please go ahead and say it loudly. I did not hear. Aman?
Yes, I'm telling that we need to find Gibbs free energy.
Yes. So if we can find out the Gibbs free energy for both of the possible macroscopic state, then we can see which one is having lower Gibbs free energy, which one is having higher Gibbs free energy. Okay, and then we can make an assessment in which state it will be found, right? So I am giving you a minute to draw all of these 5 structures on your notebook, because we are going to use this. Okay, so I will keep a silence for a minute, and please draw all of these 5 structures in your notebook, because this is something which I would like you to work on after a minute. Please draw these 5 structures in your notebook. Please draw them quickly. All right, so let me stop sharing this one. Let me start sharing my screen. So what we are given here in this problem is that there are three states which are like this. One of the states is like this, the other one is like this, the third one is something like this. Okay, and these are our open ones, right? And then the packed ones are like this. And as one of your batchmates told me that if we have to find out in which state this system will be found, we should be calculating what is the free energy of the open macroscopic state is and what is the free energy of the packed macroscopic state is, right? And this is something which can be obtained if we know the average energy of the open macroscopic state minus T into S of the open macroscopic state. Similarly, this one we can find out if we know what is the average energy of the packed macroscopic state is and what is the entropy of the packed macroscopic state is, right? Now we have to find out, so we have to find out what is the average energy of the open macroscopic state is and what is the entropy is, right? Now entropy is something which can be also written as U open minus T KB ln W open. Okay, similarly this one U packed minus T KB ln W packed, right? So here I can clearly see that how many open configurations are there. How many open configurations we have here in this problem? Anyone? 3, right? So here this will be U open minus T KB ln 3. Similarly, if I look at this one, this will be U packed minus T KB ln 2, right? What we have to calculate now is U open, average energy of the open macroscopic state, and what is the average energy of the packed macroscopic state is, right? And if I have to calculate the average energy of the open macroscopic state, I have to calculate the average energy of this one, and this one, and I have to take the average. Similarly, if I have to calculate the average energy of the packed macroscopic state, I have to calculate the average energy of this, energy of this one, and then I have to take the average. Okay. So what I'm going to tell you is how to do it for one of them, and then I leave it to you to find out the remaining ones. Okay, so let's see how we are going to find out the energy of the first one. Okay, so again let me quickly flash this slide for a second. Okay, so here you can see that the sequence which is given here is HP HH HP, and you see that all of these positions are numbered. That means if I'm starting to put the amino acids, I have to put them here in this sequence. Okay, similarly I have to put them here 1 2 3 4 5 6 7 8. I have to put them here like this. Okay, so I'm going to put the amino acids here, and then I'm going to calculate the energy. And what is given to me is that if there is a unfavorable contact, either H is right next to P or P is right next to H, then energy will increase by epsilon. If H is also making contact with water, then energy will increase by epsilon. Okay, so I'm going to use all of those hats, okay, by calculating the energy. So let's see how I'm going to do it. So the first structure is, so the sequence which is given to me is this H P H H H H H P. Okay. So first, second, third, fifth, sixth, seventh, and eighth. Okay. Remember the numbering 1, 2, 3, 1, 2, 3, then 4, 5.
Sir, you are not sharing the screen.
Oh, I'm sorry. Just let me share the screen again. Thank you. So let me do it again. So the sequence which is given to me is this H P H H H H H P, and if I'm going to start trying it, I will try. And the numbering is like this. So I have to put the amino acids like this. So this one will be H P H H H H H. Now see, this is already bonded interaction, so no change in energy. H can make a contact with water on this side, epsilon increase of energy. H can also make a contact with water on this side, epsilon increase. This side, epsilon. This one is a P, so if it makes a contact with water, no increase in energy. Okay. This one increase in energy. Second increase. Similarly, this one can make one bond on this side, one bond on this side. Similarly, this one can make a bond on this side, one on this side, one bond on this side, one bond on this side. So this one will make a bond on this side, one bond on this side. This is a P, so no net increase in energy if it comes in contact with water from inside. So 0 from this side, 0 from this side, 0 from this side. Okay. Now calculate how much will be the total energy. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So the total energy is 13 epsilon for this one. Okay. Now I would request you to find out the energy for this one and this one. Okay. What is the energy for the second one and what is the energy for the third one? Please do this. And if there are any questions, please ask. Please do this. And then once you have done it for the open ones, you also do it for the closed ones. Those who have done it can start to raise their hands now. If you have done it, please raise your hand, and don't worry even if the answer is wrong. It's fine. Tanmay, have you done it? Tanmay Ganguly.
Set.
Okay. Apurv Keer.
Sir, I've done the open one and then the attack.
Yeah. So what is the energy for the second one?
I got 13, sir.
13 epsilon. Very nice. For the second one, you got 13 epsilon. Okay. How about the third one? Did you do the third one too?
Yeah, I got 12 in that.
No, 12 is a wrong answer. Okay. So I will ask Vedika. Vedika, have you done it? Okay, we'll ask someone else. Sakshi, have you done it? Anyone else who has done it, please raise your hand. Yeah, please go ahead.
11 epsilon.
For which one? Third one?
Yeah.
No, 11 epsilon is the wrong answer. Hers. Anyone else?
13 for this as well.
Aman, yeah, please go ahead.
13 for this as well.
Okay, yeah. For the last one too, it will be 13 epsilon. Okay. Now, last two. Anyone?
Well,
No, 12 is a wrong answer.
8.
Again, 8 is a wrong answer. Make sure that you are looking at the HP interactions also. If H is within the bonding distance of a H, no increase in energy, but if H is within the bonding distance of a P, then there has to be increase in energy. So, revise that your calculations based on that. I will show you the calculations in a minute, but I want all of you to try it out. Aditya Mehta, have you done it?
Sir, is it 14E?
No, unfortunately, that's the wrong answer. Okay. Abhishek Jain. Have you done it? Please do not hesitate to raise your hand. Simran Sinha. Simran. Jay Vora. Anyone who has done it? Last two? Sravya?
Sir, is it 15?
No, 15 is again a wrong answer.
It's 11 epsilon.
Okay, very nice. Who is this?
Aryan.
Okay, Aryan. Okay, so for this one, it will be 11 epsilon. How about the second? Last one.
Sir, 11.
Okay, last one will be also 11 epsilon. Okay, now those who were not able to figure it out, I will just show you the calculation for a second before I come back to this one again. Okay. Okay, so please look at this, and if there are any doubts, please ask me. Fine. All right. Let me stop sharing this and let me again go back to my screen. So what we have found out, if you look at this one, maybe this one, for all of them, the energy was 13 epsilon, 13 epsilon, 13 epsilon. And this were the open ones. And if you look at the packed ones, you have one of the possible configurations like this. The other one is like this, and the energy of these two were 11 epsilon. 11 epsilon, and these were packed. Right. What we had written that G open was U open, average energy of the open state, minus T KB ln 3, right? And similarly, G packed was U packed minus T KB ln 2. Right? Now, you see that if you look at the open macroscopic state, there are 3 possible microscopic states, and all of them are having energy 13 epsilon, 13 epsilon, 13 epsilon. And therefore, the average energy will be also 13 epsilon. So this becomes 13 epsilon minus T KB ln 3. Similarly, in this case, there are 2 microscopic states, and both of them have equal energy, and therefore, average energy will be also 11 epsilon minus T KB ln 2. Now, we have got the expressions for the free energy for open and packed macroscopic state. I can say now that packed macroscopic state will be preferred as long as my free energy of the packed macroscopic state is less than free energy of the open macroscopic state. Or in other words, 11 epsilon minus T KB ln 2 less than minus T KB ln 3. Or we can write down, this becomes T KB ln, if I bring this one on the left hand side, enter, then this becomes T KB ln 3 by 2 less than 13 epsilon minus 11 epsilon, this becomes 2 epsilon. Okay. So now, if you look at the origin of this term, this is coming because of the differences in the average energy of the 2 macroscopic states. Similarly, if you look at the factor of this 3 by 2, again this is coming because of the differences in the entropy of 2 microscopic states. That means, and if you solve it further, what you are going to get is that 2 epsilon, the T, if the T is less than 2 epsilon divided by KB ln 3 by 2, then the packed macroscopic state will be preferred. That means we have some transition temperature T star which is given as 2 epsilon divided by Kb ln 3 by 2. And look at how this transition temperature depends on the properties of both the macroscopic state. This factor of 2 epsilon is coming because of the differences in the average energies of the two macroscopic states, and this factor 3 by 2 is coming because of the differences in the entropies of the two macroscopic states, and therefore this transition temperature is being determined by the competition between these, the differences in the average energies as well as differences in the entropies of the macroscopic state. Okay, and this is how we can apply the idea of free energy minimization to understand what will be the shape of the protein. So what we did here, we used a very simple model of protein folding, which is also called HP model of protein folding, to understand under what conditions the protein will be found in different macroscopic state. So here we took a simple example of open and packed macroscopic state. All right. All right. So this is the summary of what we did today. We looked at proteins and their structures, and we found that proteins are free energy minimizers. We looked at the concept of microscopic states and macroscopic states. We looked at the relation between the free energy, enthalpy, and entropy, and we also looked at the relationship between the free energy and the partition function. We looked at the expressions for the thermodynamic entropy as is equal to Kb ln W. Then we also looked at the expression for another alternative expression for entropy, which is called Shannon's entropy. And finally, I discussed a simple model of protein folding, which is also called HP model of protein folding. All right. So what I will do, I will stop here. I will stop the recording, and then I will take any questions that you may have. All right.